Hi, thank you. I'm very excited to talk to you about how to model longitudinal neuroimaging data. It's something I've been passionate about for quite some time. Okay. I'm going to just start with some ingredients, some basic ingredients for measuring longitudinal change. Some of these are fairly obvious. You need longitudinal data. You need a sensible measure for time. But what I think may not be so obvious to some is that you need enough occasions of measurement to examine the correct form of growth. So if you're interested in how people are changing, that trajectory of change, not just the absolute amount of change, you need enough occasions to be able to model that slope or that curvature. And that means that you need at least 3 occasions to look at linear growth. And in general, and that's because if you just have 2, you're confounding the measurement error of your measurement along with the individual change. So basically, more measurements that you have, the better. But the focus of this talk is largely on you also need the right statistical models. This is just an example of what I'm talking about when I speak about different individual trajectories. If each light blue line is an individual person, you can look at their change across time, and you can think of this as being linear or cubic or quadratic or anything in between. These are some questions that we might want to ask about change. First of all, once you understand the form of change, you might want to then know does that differ across individuals. We might then ask, what is the relationship between change and time-invariant predictors? So for example, do people with a specific genetic subtype have a steeper slope of decline in Parkinson's disease? We then might ask, what's the relationship between change in some outcome measure and change in a predictor? So does change in functional connectivity relate in some way to change in cognition? We might ask, are there latent groups of individuals with different change trajectories? Maybe we can find some sort of latent groups of pathological change that manifest themselves in different trajectories. And we can ask, the last question I wrote here is just, does the trajectory of change predict outcomes? There are 2 statistical frameworks I'm going to talk about today that allow us to answer these questions. And the first is mixed effects models, also known as multi-level models or hierarchical linear growth models. And the second, in red, which is latent growth curve modeling. So this is a more flexible superset of multi-level models, but it is actually a subset of the larger class of structural equation models. And structural equation modeling is a very, very powerful framework that allows you to examine quite a lot of questions about change, and I'll talk about why that is. The most important thing here is that of these questions that I listed, which all seem sensible, only the first 2 can be answered with mixed effects models or multi-level models. The ones in red need to be answered with a framework or best answered with a framework that is a structural equation modeling framework. And this is sort of the point that we came to in some of our longitudinal studies, where we're like, well, we'd like to be able to do this, and we can't, because right now, fMRI analysis is really limited in the mainstream to mixed effects models. So just to introduce some terminology that you'll hear about later, mixed effects models, the mixed part of that refers to the fact that we have both fixed effects and random effects. Some examples of fixed effects are things like male or female or treatment or control. Random effects, subjects with fMRI data or subjects with multiple measurements. We assume with fixed effects that all the groups that we care about are sampled. Whereas in random effects, we assume that not all groups are sampled. The groups come from a probability distribution. With fixed effects, we care about those groups only. And with random effects, we care about generalizing from those samples to a larger population. And with fixed effects, we model the population mean, and random effects, the variance. And Dylan will talk more about mixed effects models in his talk. But as I mentioned, the state of the art in fMRI analysis, I should say mainstream fMRI analysis software, FSL, SPM, is that they implement a multi-level model as a 2-stage analysis and don't handle a lot of things, a lot of cases that you need for true longitudinal analysis, missing data just being one of them. AFNI is probably the most creative in this way in that it implements wrappers around the R NLME library, which is a full mixed effects model library, and gives you quite a lot of power to do a lot of things, and that's very good. But none of these allow you to do the latent growth curve modeling, which can allow you to answer these questions that are so important. And the reason that latent growth curve modeling is so powerful is because you have this distinction of a latent variable, which is a true random variable, but it's hidden from us. So, for example, the slope of change is something you don't actually measure. You measure some other thing that you're looking at across time, but then you have to fit a slope. And so those are indicators, those measurements that you take, and then the latent slope and maybe intercept are latent constructs. So once you have these, then you can model the relationship among these latent variables, and you can look at them as predictors of things you care about. You can look at what other things predict those. You can look at change in two variables and see how those are related. And it gives you a lot of power to answer those questions. So basically, using this framework, you can formulate those research questions about change in a structural equation model. This is an example of just a linear latent growth curve. We have 4 occasions of, the way that it's typically shown here is that in boxes are those indicators, the things you actually measure. So, and then in bubbles are the latent constructs. So you can see here that you're measuring something at a baseline 1 year, 2 years, 3 years, but then you fit a latent intercept and a latent slope to that data. Now, this is something that you can do with a mixed effects model. But what's powerful is that you can go ahead and go crazy with these sorts of things. So this is taking 2 such growth models and combining them. And this is just a behavioral example where on the top, we're looking at a growth curve of depressive symptoms, and on the bottom, we're looking at a growth curve of post-concussive symptoms. And what we care about is how those slopes and intercepts of those things relate to each other. And that gives us some understanding of the trajectories of change and how they are intermingled. So if you find yourself thinking, well, I want to take these kinds of models and I want to implement them using my fMRI or other neuroimaging data, well, you're in luck, because we were in that sort of situation. We thought, well, how can we do this? And so this motivated the design of Neuropointillist, which is just an R package, and R because R is very much a vehicle for cutting edge statistics. And so that's why we chose it. The statisticians like R, use R. You can communicate with them through R. But the basic idea is that you take your fMRI data, you pre-process it, and maybe even do the single subject analysis if you're looking at task data, as a lot of us are. And then you run the Neuropointillist package, and because we're limited to GLM here, or not, well, we're limited to a voxel-wise analysis. So basically what we're doing is we're trading off the fact that your modeling in R is going to be a little bit slow, but we have this massive parallelism. So computers are cheap. We can buy compute time right now on cloud services for very, very little, and certainly for much less than it would cost to hire a programmer to optimize your code or take your time to optimize your code. So we get some speed up by basically just doing this massive parallelism. This is just a little bit more detail on the approach here of what the software package does. You give it data sets, and you give it sets of covariates and a mask, and it just flattens those 3-dimensional, 4-dimensional data sets into a structure that is your voxel data and your design matrix, except that the columns are named as your covariates kind of coherently. You provide a model, and that defines a process voxel function. So you can put any model in there that you want. And as long as you give names to the things that you return out of that function, it will use those names and generate output files with those names. So the sky's the limit, and this is probably the most confusing thing about it, is that you can do any sort of modeling you like in there at all. You have your choices then of doing parallelism on a multi-core machine, or my favorite is that you can output chunks of data and to process and do that on a cluster, essentially. So I think I've been scarred for life for having to literally move processing chunks to different machines that aren't clustered just to get things done in time. So I always leave that as a backup. I'm sorry, let me just, to make one more point about this. This is a really simple package. I mean, nothing I described here is difficult to do, and it's very straightforward. The power here comes from this ability to write the function that you want in R. Once you've written your model in R, That's something that you can share with other people. Statisticians can read it. You can give it to another lab so that they can run the same statistical analysis, assuming the pre-processing is coherent in both labs. It's very, very reproducible and transparent to do that, and very simple to do. And this has been very powerful for us. This is just an example to prove to you that you can actually get things out of this crazy approach. This is a work that my student, Brianna Lee, my undergraduate, Brianna Lee, did, looking at data from the ADNI data set. And what she did was look at functional connectivity at rest from the posterior cingulate cortex to the rest of the brain in 3 groups: normal aging, mild cognitive impairment, and Alzheimer's disease. And what is striking about this, what you're seeing here is the slope, whether it either increased with time or decreased with time to the PCC. And what's striking about this is that the decreased anti-correlation of the attentional network and the default mode network that is often attributed to an aging effect, and I, in fact, have attributed in the fact to an aging effect. You don't see that in the PIB negative normal aging group. It is only in the MCI group. So this illustrates, I think, some of the power of longitudinal data analysis to separate out these trajectories that we kind of lump in together when we do cross-sectional types of analysis. This is where you can get the software. This is the IBIC GitHub repository, and I have a tutorial there, and please feel free to visit that. And finally, I'd just like to thank all of the collaborators and students and other people who've really helped so much at the University of Washington and the University of Oregon to make this a reality. Thank you.